My name is Brad Nickerson. I'm president of the Jemseg Grand Lake Watershed Association. Our watershed covers 3,950 square kilometers, which is more than 5% of the area of New Brunswick. And I think it's around 70% of the area of Prince Edward Island. So it's a pretty large watershed. And Grand Lake is the largest freshwater lake in the Maritimes, 171 square kilometers. And if you're not already a member of our association, we'd love for you to join us. Memberships cost $10 per year. They support our activities, including the organization and hosting of this webinar. And all you have to do is click the Join Us button on our website. We welcome your questions during the webinar, and we would prefer that you use, well, it's the only real way to communicate with us, is to use the Q&A question and answer window to ask your questions. And just type your questions in as they come to you, as the questions arise. And I will relay those questions to Dr. Bruce at the appropriate time. So if you want to ask questions, please feel free to type them into the Q&A window, and we'll make sure that Meghann sees them and can answer them, or hears them, I guess. So we welcome your questions. So please feel free to ask questions by typing in the Q&A window. All right, so we're going to have a quick little poll now to ask a few questions of you. Actually, one question that you'll see in a moment. It's going to be a live poll, and the poll is to give Meghann Bruce a sense of who her audience is. So there's going to be a question come up on your screen here. I'm going to share it right now. And if you could click one of the answers to that question, do you know what the word benthic means and how it relates to cyanobacteria? Just so click one of those answers that you see there. There's 4 possible answers, and that would be wonderful. We'll let it go for about another 20 seconds or so, 20 to 30 seconds. And as soon as that time expires, I'll just share the results of that poll with Meghann and with the audience. Okay, so how are we doing here? 80% have answered it already that are attending at the moment. Maybe I'll wait till it gets to 85. Can we reach 85? It's just like COVID-19 vaccines. May never get to 90, but at least we'll give it a whirl. Okay, we're at 86. I'm not sure what Premier Higgs is saying his target is now. Okay, we just crossed 90%. I think that's pretty good. Okay, I'm going to end the poll and we'll show the result here. And I'm hoping Meghann can see this. Oh yes, sorry, here it comes there. I didn't needed to press one more button. Can you see the result now?
Yeah, I see it. About half of us have no idea. 30% do and can explain it to others, and 20% have heard the word before but don't know how it relates to cyanobacteria. And so we're a very mixed background tonight, which is exactly what I teach at university. So that's great.
Okay. And nobody thought to affiliate it with Ben Affleck. That's a good sign, I think. All right, so I'm going to stop sharing that. Okay, so we're going to proceed now into Dr. Meghann Bruce's talk, and I'm going to introduce her. We're very fortunate to have Meghann Bruce with us this evening. She's a very experienced researcher and also has a lot of field experience, which she's going to share with us, some of that, which she's going to share with us this evening. She's a research scientist with the Canadian Rivers Institute, and she received her PhD degree in biology from the University of New Brunswick in 2015. She's a naturalist with a keen interest in aquatic ecosystems, and her research projects emphasize the application of science-driven biomonitoring initiatives to support aquatic ecosystem managers and decision-making processes. Her current research projects include seeking to understand the environmental conditions that led to the recent appearance of toxin-producing cyanobacterial mats in the St. John River, a fairly new phenomenon as I understand it. In addition to her research, Meghann teaches undergraduate courses and field-based workshops in the fields of botany and conservation environmental biology. So Meghann, over to you, and I'm going to allow you to share your screen here. I think you should be able to share it at the moment.
Let's see. Great. Thank you, Brad. Everyone should be able to see my screen now, and I'll just switch it into presenter mode. Are we good?
That looks good to me. Any issues come up, please correspond by typing into the windows, the Q&A or the chat window. I should be able to see them both.
Great, and I'll just rely on you to watch those because I won't be watching them.
Okay, very good.
Great, thanks. Thank you all for coming tonight. Your presence here tells me that you are interested and engaged in the issues that are facing our freshwaters in New Brunswick. And as someone who is very passionate about what they do and very dedicated to their research, this is very encouraging to see. So thank you for your interest. I look forward to your questions and the discussions that we will have. So please don't hesitate to ask questions throughout this presentation. This is like a story. This is a science story before we go to bed tonight. And I want you to ask questions so that the story is complete for you. I may not be able to answer all of them, but I can give you what we know so far. Today, we're talking about cyanobacteria, and we're focusing on identification of cyanobacteria for the purpose of keeping you and your pets safe. My work on this started in 2018. I've been working with the Canadian Rivers Institute since 2014. But in 2018, I collaborated with other researchers to investigate the appearance of blue-green algae, also called cyanobacteria, in the Woolastook. And this was a direct response to the dog deaths in 2018 and another in 2019. As you can imagine, this is very tragic for those that experience this going through their daily routine, walking their dog along the river. They notice something is wrong with their dog, and within half an hour, their dog is dead. And then when this makes it to the media, it's very concerning for the public, because other than hearing that dogs died of cyanobacteria or blue-green algae toxins, it's not the whole story. It's what are the blue-green algae? What are the toxins? Where did they come from? Our river looks normal. So how do we explain this? Because we hadn't heard of this happening before. And for me, this was concerning because I had research teams on the river, about 12 summer students, technicians, grad students. There were about 20 people on my research team on the river every day. We're running usually about 6 field teams. And some of those people were snorkeling, doing mussel surveys, fishing with seine nets. So we needed to know what it was happening in that environment and what was causing this. So there were multiple reasons and motivations for getting this work started. And like I said, it's a collaborative effort of various expertise, and my role in this is the field programs. So that started in 2018, meaning we now have 3 years of data and observations. We had to start from scratch though, and so we had to figure out what were the cyanobacteria, what were the algae in this region. And today I'm going to start very general with this group, talking about what are cyanobacteria, benthic cyanobacteria specifically. And in a moment, I'll clarify the terms blue-green algae and cyanobacteria. Then we'll look at identification for potentially toxic cyanobacteria. It's important to point out that when I say identifying potentially toxic, that's the best we can do because we can't tell if something is toxic or not by looking at it. So what we do is we identify cyanobacteria and change our behaviors accordingly to make sure that they are safe. And as we go through this presentation, I'll be sharing what we've learned about cyanobacteria blooms in our region and hopefully answer some frequently asked questions or explain why we still can't yet answer some questions. So cyanobacteria are, like the name suggests, bacteria. The term blue-green algae has been applied to cyanobacteria, so they're essentially the same thing. But the problem with the term blue-green algae is the word algae describes a whole wide range of organisms, including, for instance, marine algae, like kelp, are also algae. But cyanobacteria are the algae that are bacteria. They're the only bacteria that are blue-green algae. So the terms you may see used interchangeably in public communications, but I like the term cyanobacteria because one, it reflects what these organisms are, they're bacteria, and it puts less emphasis on the term blue-green, which by the end of this presentation you'll see is somewhat misleading. The term blue-green was applied to these bacteria because they are blue-green in color under a microscope. What you're looking at here is an image of under the microscope of colonies of bacteria. You're looking at chains of cells. Within each one of these lines or these filaments are multiple bacteria. Each one can break off and start a new chain. So they're filamentous bacteria in this particular type, but there are some types that are just individuals. These ones are what we call colonial. And notice they are blue-green in color, but that is not always the case. These bacteria grow in aquatic environments, but also damp environments. They're virtually ubiquitous in the environment in that they exist in soils, they exist in pores, in rocks, around the area of tree roots. They also live in hot springs and have been found deep in Earth's crusts. So they're ubiquitous in the environment. And there are many different kinds, just like we use the term birds to describe many different species of birds. When I say cyanobacteria, it could mean a various array of different organisms. So cyanobacteria is a general term, but know that there are different types. And despite their reputation, they're actually very important to ecosystems. So they're green, blue in color, like you notice, and that's because they're photosynthetic, like plants. They fix carbon from the atmosphere and they generate oxygen. So they oxygenate waters. They've also are what gave Earth its first atmosphere. And because they fix carbon, they also are a nutrient source for other organisms. So they form these mats or what are called biofilms, and other smaller organisms feed on them as well. So they have several important ecosystem roles. But sometimes, and this is likely why you've heard of them, sometimes they produce toxins. These toxins are called cyanotoxins because they come from cyanobacteria. And there are many different types. There's hundreds of different types of toxins, just like there are hundreds of different types of cyanobacteria. And we group these toxins by their targets, what the toxins do. There are hepatotoxins, which attack liver and kidneys, neurotoxins, which are neurological, they affect the nervous system, and dermatoxins that irritate skin. And even within these three main categories, which are shown on the left of this chart here, there's several other types of toxins. The top one you can see here is called microcystins. And even that is a group. There are many types of microcystins. Colleagues of mine in Nova Scotia discovered over 180 different types of microcystins last year. So we're still discovering new types of toxins all the time. And these toxins don't always occur in isolation in cyanobacteria. Sometimes there's multiple types of toxins being produced simultaneously. I often get asked the question, why do cyanobacteria produce toxins? And this is something that scientists don't yet know. It may be just a secondary byproduct of metabolism through something else that they're producing. There's some evidence that it may affect chemical processes that help them be competitive with other bacteria or other microorganisms in the environment, or affect their availability for nutrient cycling. So we don't yet know why cyanobacteria produce toxins, and we don't know what triggers this toxin production. So sometimes cyanobacteria can be not toxic and then become toxic in a bloom. So there's lots of variability surrounding toxins. And when I'm talking about blooms today, I'm mainly emphasizing our research on benthic blooms. So there's two general types of blooms. You can have a planktonic bloom, which is when these bacteria grow throughout the water column, floating around, giving rise to this green pea soup color. This is what we call a planktonic bloom. It's like a plankton. And a benthic bloom, conversely, grows on the bottom. That's what benthic means, growing on the bottom of the water, of course. So right on the rocks or the soils or the sediment, the substrate. And a benthic bloom, notice in this bottom picture here, the water is actually clear. What you're looking at, that brownish gold color, is like a carpet of cyanobacteria that is growing in the benthic environment. So that's how a benthic bloom differs than a planktonic bloom. Both of these blooms have the potential to produce toxins, and in variable amounts. And different types of bacteria can come together to form these blooms. So when we're looking at this planktonic bloom, for instance, here on the top, while there may be one abundant type of cyanobacteria, there's chances are there's multiple types in that bloom, and definitely as well for the benthic blooms as well. Another question I often get asked is, what causes cyanobacteria growth? And there's not one simple, straightforward answer to this. Rather, it's a suite of environmental characteristics. First of all, they need light. Like I said, they're photosynthetic like plants, they need light. This is why we often see them growing in shallow waters or deeper if the water is clear. They also grow in both still waters and flowing waters. So water velocity is important for different types of blooms. You've probably heard the advice, watch out for slow moving stagnant waters because of cyanobacteria or blue-green algae. Now, that's not untrue, but it's also true that they can grow in very high flows. Our work has shown that some of the highest flows we have here on the river are where our toxic cyanobacteria mats are proliferating. We also hear temperature quite a bit, and temperature does influence growth, but again, it influences different types of cyanobacteria differently. It's a bit of a misunderstanding that cyanobacteria only proliferate in late summer when waters are warm. Yes, that's true for certain types, but the type I've been studying shows up, it's there as soon as the flood retreats and I can safely get to my field sites the end of May. And by June, they're very well established. So temperature isn't as much of an influence, or isn't equally an influence on all types of cyanobacteria. And you've probably heard nutrients. And nutrients do affect all living things to some degree, but especially the benthic cyanobacteria, so the ones that form that mat on the substrate, on the bottom of the water. Those bacteria are really great at getting nutrients, even in low nutrient environments. So for instance, nitrogen and phosphorus are two nutrients that have been loaded in our aquatic ecosystems that have caused what we call eutrophication, so lakes turning green. That is the outcome of planktonic bloom. But in the benthic environment, even without nutrient loading, just natural levels of nutrients, these bacteria have geochemical cycles that make them really great at taking up even limited nutrients. So nutrients aren't always an indicator of what will trigger a bloom or not, but it certainly plays a role as well as other biotic and abiotic influences. So there's a lot of things that cause blooms. These things that cause blooms are different in different areas. So each bloom and each, especially a problem bloom, needs to be studied individually. So the question we're here to answer today is, how can we identify potentially toxic cyanobacteria? And this is where it all started with our research. When those dogs died and everyone was wondering what has caused this, besides we know it's blue-green algae, where is this blue-green algae growing? How much of it is there? when is it growing? These are the questions we wanted to answer because we wanted to figure out what blue-green algae was contributing to this. Was this going to be a problem that we see continue? Was this a one-year thing? What we still don't know is how far back this goes. We think of this issue as something that just appeared, that just showed up, but it's possible that this has been an issue for longer than we think. We just didn't realize it, especially if either dogs hadn't died, the algae could have still been there and not ingested. Or conversely, if someone was walking an older dog along the river and thought that their dog just took a seizure or died of natural causes. So it's really hard to say how far back this issue goes. So we were starting from scratch. It's really hard to design a biomonitoring survey when you don't know what the thing is that you have to collect. So when we started off, we collected everything that we could find. We spent many days out on the water with our team covering the Fredericton region was our first study region. We've since explored all the way up to Edmundston and this year downriver, looking for blue-green algae, what types were growing, when they were growing. And to do this work, we partnered with Dr. Janice Lawrence, who does our genetic test to see if the organisms we're collecting have toxin-producing genes, and the National Research Council of Canada Metrology Lab, so the biotoxin lab in Halifax, as well as many others. There was tons of other groups that had volunteered time and eyes on the water to increase our ability to survey a wider area. And like I said, we surveyed everything because we didn't know what exactly was producing this toxin. So we started scraping rocks, like the top that you see here, with light colored slime, rocks with dark colored thick slime. We sampled scum on the top of the water. And in the right-hand corner here, you can see aquatic plants. We wiped down aquatic plants for all of the organisms that were growing on them because maybe this was something that was growing on plants. And in the bottom right-hand corner, there's actually two things in this picture. There's the green thing that is filamentous and then the green bubbly thing. We were sampling every different looking thing that we could find. And we were screening it for toxin-producing genes and to see if those genes were being expressed, to see if it was actively producing the toxin. And now let me show you what we found. We found that mats, cyanobacterial benthic mats, start growing as early as we can get out in the season. They are there the end of May and very well established by June. At the start of the season, this is what they look like. See this patch on the rock that is covered with bubbles? Those bubbles are oxygen from the cyanobacteria photosynthesizing. This is a very thin mat, and our analysis have shown that the toxin production starts as early as May, June. At this point, these mats are only a couple millimeters thick. They are thin and bubbly, and they do tend to be dark green at this stage. So the name blue-green algae isn't totally misleading at this point. But within a matter of a couple of weeks by June, mid-June or July, things change. Those mats, notice here in this first picture, you can still see all of the gaps between the rocks. But then by mid, late June, early August, we see that the mat grows over the rocks. In this picture here on the left, this is actually a pile of rocks in the substrate, but it looks like mud because what that mat is doing is it's growing across the rocks and connecting them. Also notice that in this picture, there are multiple colors. There are this beige color, there's this black color with the stuff peeling off of the rock. This is all cyanobacteria. And in fact, what's not cyanobacteria in this photo is that bright green part that you can see emphasized here on the right photo. The part that's bubbly in the photo on the right is cyanobacteria. But then behind that in the photo, that bright green, like everyone thinks is blue-green algae, that's actually just green algae. So when we launched the study and asked people to go out and collect specimens for us, we were getting a lot of specimens that were green algae that weren't necessarily blue-green. And this is because by midsummer, a lot of the green color had faded And we're seeing a lot more brown and beige. Notice how these mats are peeling away from the rock. At this point, they're about a centimeter thick. They're very smooth, peeling. They almost look like, and it's a disgusting way to describe it, but my students have said it looks like rotting flesh peeling away underwater. And yeah, the color is extremely variable, anywhere from dark green to beige, anywhere in between there. So this is an example taken June 10th this year on the Woolastook. Notice the webbing between these rocks. What you wouldn't recognize right off the bat is this is actually a photo taken through the water, from above the water. So this is probably about 6 inches deep, maybe a bit more, but that water is crystal clear. And cyanobacterial mats can actually proliferate in very pristine waters. They're also present in some Arctic streams. They're present in many other places in the world as well. New Zealand is having problems with cyanobacteria proliferation, for instance. We've modeled a lot of work of what we're doing off of the issues that they've encountered and their approaches to studying them there. But this mat is June 10th. It is clearly very well established. By no means is it our hot late summer conditions that we would expect. And again, the green in this algae actually isn't cyano. That rock that's dead in the center, that is just green algae. And the cyanobacteria is the beigey brown stuff all in between the rocks. And this webbing and this coating between the rocks is key. Then by July, August, what happens, sometimes the water levels drop and come back up. And if these mats are exposed to air, especially, they turn this golden color. They're orange. Also notice, looking at this mat, you can see large bubbles in this mat. That's air, oxygen trapped in the mat. And again, the green in the background, this is just green algae. At this point, they're still about the same thickness as they were in July, but their texture is starting to change. They're spongy and lumpy. You can still see the air bubbles, and their color has changed yet again. At this point in the year, it is still possible to see some of the mat morphologies that I showed you previously, if that mat was just recently established. But in the regions where we're measuring our mats and looking at them, they tend to look like this golden brown. And what happens next is all of those air bubbles start to cause this mat to lift and peel up off of the bottom. So in red here, I've drawn an outline, hopefully you can see it, of a mat that is starting to lift off of the substrate. And this is one way you can identify a cyanobacteria mat. If you see a substrate with a carpet like this and you kick it with your boot, it will immediately float to the surface. It may be pulled down, if you're in a flowing environment, maybe pulled downstream a few meters first, and then it will float up and float along the surface. This is what we call liftoff. And this is a stage in the life cycle of mats where they can distribute beyond where they're just growing. So one of the things we don't emphasize when we're sharing our research is the exact location the mats are growing, because that's not necessarily the highest risk zone. Most people wouldn't encounter these mats when they're growing on the bottom of the river as much as they would when the mats lift off and can float downstream on the water surface. So here's an example of a mat floating on the surface. You can see, if you look down through the clear water column, this is probably close to, I want to say 75 centimeter deep water, again, crystal clear water. This mat has floated up and will now flow down river to wash up on a shore. And this is where dogs were encountering the mats, was these wash up zones. When this happens, where this mat ends up depends on the flow of the water, but also things like wind. Wind can blow these mats inshore. So it's really hard to predict. We're watching wash up zones, but it's hard to consistently get a feel for where things regularly wash up or how far downriver they can go. When they wash up, they can, again, look totally different. They can look like this orange goo here, the rocks, or brown. This is brown floating in with a bunch of plant debris. And this is the tricky one here in the top left. This is gray and dried. This is the matter that is thought to be what the dogs were ingesting, according to the owner's observations. And although this mat is dry and the cyanobacteria mat is no longer alive, there's still toxins inside that mat. So these mats can hold those toxins and carry them to other locations where they wash up on shore, dry out, and just sit there with the toxins in them. So this is why we want people to learn to identify them so they can avoid them and look for things like this on their shorelines. So here's an example of what a cyano mat site may look like. We do see them growing in smaller patches, but some of the patches that we're seeing in the Fredericton region are hundreds of meters. So they're very large, extensive mats. And one of the first cues that got me looking at the area when we first discovered these was this greenish tinge along the shoreline. So the water level here has dropped and there's this green tinge. And this was taken, I think, early June. And a couple weeks later, there is mats. So they grow very fast and they then connect the rocks and form this webbing. I will also say that when we see mats show up, what we're seeing between years is they consistently grow in the same area. So there's something about those habitats that are causing mats to grow in those locations. Here's an example of what a mat site won't look like. I get a lot of calls about this, and this is just regular green algae. It was taken by Aaron Fraser. I think he was working with the Armucto Lake Watershed Group, Armucto Lake Association, sorry. And this is a Spirogyra bloom. This is a filamentous green algae. So it's very, very green. But because the term blue-green is so widely used, I can understand why people think that this is blue-green algae. I mean, I still wouldn't want to eat those waters or drink those waters for other reasons, but they are not cyanos. So what to look for with all of these range of morphologies, if you take anything away from this, what you need to be looking for is the bubbles, these tiny little bubbles and the ability of these mats to peel and float. So color can be variable. I wouldn't put too much weight or emphasis on color for what they look like, but the texture, the bubbles and the floating are things to look out for, knowing that these change throughout the year. So common advice that I see given for avoiding blue-green algae or cyanos is look out for slow-moving, stagnant waters, waters with odor or discoloration, and algae that is blue-green in color. When our research has directly contradicted this, it's not that these aren't important points for other reasons, like planktonic blooms in some areas. But there's more to the story than that, in that we find cyanobacteria many different colors, from golden, green, brown, and they are growing in some of our fastest flowing clear waters with no odor. I say no odor. To me, all lake water has some odor, but no noticeable odor. It's not a rotten smell like when you're visiting a mud flat or something. And they're certainly not always blue-green in color. So it's not as simple as looking out for those areas. We need to learn how to identify these and recognize that they have the potential to not only be in the areas they grow, but washing up on shorelines. So a question I normally get at this point is, how do we make cyanobacteria go away? And the unfortunate part is we don't. Once a system has mats or planktonic blooms, they tend to reappear. These mats that we're finding in the Fredericton region of the Woolastook, New Zealand is facing similar types of mats, and they've been dealing with these mats for about 15 years. And if anything, they've only found that they tend to spread. So the best way to manage this, the safest way to manage this, is to manage human behaviors with the mats. And it's as simple as identifying what they look like and avoiding direct contact and ingestion. It doesn't mean that we can't enjoy our recreational waters. I still go out on the river, and our research teams are certainly out in the river. We're working in areas of the mats every day. We're just a little bit more careful when we're handling things, and we make sure we don't bring our dogs to the field with us. Another question I often see, this is one I see on social media, with increased awareness of this issue, it's fantastic that more people are observing and reporting. I often see GNB criticized for not doing more sampling. But it's important to know that there is no amount of sampling that they can do to guarantee safety. And this is because of the variability surrounding cyanobacterial blooms and the toxins they produce. So for instance, there's variability in toxin type. I said there was hundreds of different types of toxins. To do a test, you often have to know what type of toxin that you're testing for. You have to know what it is that you're testing for. The other challenge is spatial variability. Brad, I forget how big you said Grand Lake was. I know that it's a very large lake, but toxin variability changes even within a bloom or across a mat site. So you may collect one sample that is not toxic. Meanwhile, a few meters away, another sample could come back toxic. And then a few meters past that, it may have 10 times less toxin in it. So there is the spatial variability in toxin. And the other aspect is a bloom temporally changes with its toxin production. So you may sample a bloom in the morning and that sample may appear to not be toxic. And then by the end of the day, it could be toxic. These organisms could start to produce the toxin. And there's no such thing as real-time toxin analysis that will cover all of these scales. So there is no amount of screening that can guarantee safety, but what can guarantee safety is your own eyes on the site at the water of your own beaches and learning how to identify these and control your behaviors or your dog's behaviors when there is a threat present. So the good news is that we do now know what to look for. 3 years ago, when I was in meetings designing this research, they said we didn't even know what to look for and we didn't know what to tell people to look out for, but now we do. So we are continuing to share this information. It seems like very basic information for what something looks like. It's very simple research, but a lot went into figuring out what not only was blue-green algae or cyanobacteria in our region, but what were the toxin producers. And just because we had a toxic sample once, was that consistently holding up? So it took many, many samples over our 673 kilometer long river to be able to figure this out. But the good news is we have, and our research from there is only growing. I guess I'll talk about future directions in a moment, but we are very lucky to have the team of researchers that we do here and this collaboration of expertise, because a lot of regions facing these issues don't have that. So I'm really thankful for my colleagues that I have to be able to support this research. And the continual sharing is so important. So things just like being here tonight and having all of our watershed groups engaged to help us with this, because it's a lot of work to organize these types of seminars. So I appreciate when I can just show up and talk. We also have partners like ACAP St. John that has made an informational video and pamphlet to share. And this work really is ongoing. I can't emphasize that enough. When we feel like a broken record that we've said it a million times, there's always something that checks me in to help me realize how much work we still have to do. Last year or the year before, I guess it was out on the river and we were going out to monitor our cyanobacteria sites. And there was a kids group playing down by the river. And we're there launching our boats. And I knew that this had been a wash-up site before where we had seen cyanobacteria mats wash up. So I couldn't just get on the boat and go away without checking the site first. So this kids camp, all dressed in their matching shirts, is playing at the water's edge. And a little guy who's about 5 years old is holding up a stick. And he's rubbing this stick in this little girl's face saying, blue-green algae, it's yummy, you want some? Meanwhile, his supervisors on their cell phone standing over at the water's edge, making sure the kids don't drown. But that person probably wasn't trained to identify what mats look like. Now, luckily, the kid didn't actually have blue-green algae or cyanobacteria mat on that stick. He had Eurasian water milfoil, which is another issue in this area. But his parents had obviously told him something, knowing he was going in the water and to be careful. But that is typical kid behavior. And those are the types of things we want to try and watch for when we're around the water. As another example, our team was launching, it was maybe about a month ago, maybe not quite. And as we're launching our boat, we're loading all of our equipment, and we've got all of our probes and measuring devices. And a guy comes down to the water's edge with his dog. And again, it's an area where we've seen cyanobacteria mats wash up. So we just start talking loudly about cyanobacteria mats, and he pipes in and he says, "Oh, it's fine. Just don't let your dog drink the water." And here I am with the team of researchers that are going out to study these mats, and not one of us knew really what to say or react. It's not necessarily the water, but the ingestion of the mat itself in that region. And so I do regret not speaking up. I guess I was just kind of caught off guard, and I didn't really know what to say when I was having my research explained to me. Maybe I don't, I guess, need to do the research if the questions are already answered. But that told me that we still need to be sharing. We still need to continue this process, even if it feels like we've put the information out there. We need to not only share what we found so far, but continue to update so that we can help people understand what's happening in our region. And thankfully, like I said, we've got the team to continue this. Our team continues to grow. We've got new collaborations forming in the next year. So now we know what to look for, and we know what to sample. We're starting to look at things like biogeochemical cycling and nutrient influence. So there is a new professor in the faculty of University of New Brunswick, and her work, she designs microprobes. So we're going to be collaborating, and she's going to be leading the nutrient study for cyanos in our region. And with the Macrophyte aquatic ecosystem study, so this is a study that has a research intensive area below the Macrophyte dam down to, well, downstream. As far as the Westmoreland Street Bridge, at least, we have a very high resolution hydrodynamic model that has been formed for that study. So we can look at things like water depths, substrates, flows, and temperatures to start to model these habitats and figure out why we see cyanobacteria continuing to grow in the same areas year after year. And we're also looking at the life cycle of mats. So do these patterns hold up seasonally? Do we always see the growth patterns that we do and that maturation and liftoff of mats at the same time of year? You'll notice usually around mid-July, there's more press releases and more media coverage of the cyanobacteria mats. And that's because we're trying to remind people of this issue so that they don't forget from year to year. We're also working on new tools for biomonitoring, such as using drones for data collection. When we started this work and we were sampling everything, we were getting little bits of things from the river until we discovered these massive mats that are hundreds of meters. And we did map them with transect work, but now we're thinking that drones will provide a much higher resolution for the spatial coverage, so we know the extent of these mats. We're also using something called SPATS, which some of your watershed groups may have helped out with, which looks just like a tea bag that we put in the water. And that's for toxin detection. The idea being that if we detect toxin in the water, but we can't find a mat, we know that there's a mat or a bloom around somewheres. So that is a cue for areas where we need to look further. And we're continuing to develop capacity for more analysis. So one of the limitations for doing work like this is the processing of toxin samples and the access to facilities to do this. But this is expanding, and there's another faculty member at the University of New Brunswick who has, received, I think, an NBIF grant where they are increasing our capacity to conduct anatoxin, at least, analysis, and potentially other cyanotoxins. And again, the other part of this, as I've said, is just continued dissemination, in which we rely on our government partners, our NGOs, rights holders, and stakeholder groups to do that. So when I say this next part, what should we do? This is what I hope that you can take home and remember from this. The best thing that you can do at this point is learn to identify cyanobacteria. I will have Brad forward a couple of resources to participants to help facilitate this. And once you identify them, you can then engage in recreational activities safely, meaning you avoid direct contact with cyanobacteria mats. Doesn't mean that you can't go boating, doesn't mean that you can't go fishing or paddling. I still do all of these things. I just don't handle mats. And honestly, most people wouldn't want to handle mats anyway, because they don't look too appealing. It's things like kids at the water's edge playing that we want to be able to look out for mats and to avoid them. So avoid direct contact with mats and avoid drinking fresh waters. I mean, you should do that anyway. Cyanobacteria aside, you shouldn't be drinking fresh waters, and avoid letting pets drink from lakes and rivers. Note that dogs are also particularly very attracted to these mats. That's why it's been problematic for dogs in particular. They will seek out and smell these mats and ingest them. And as a pet owner, if you have a pet, I'm sure you know how hard it is to get something out of a dog's mouth once they've decided that they're going to eat it. And the danger about mats compared to planktonic blooms is planktonic, you have to drink a lot more water to ingest the same amount of toxin. But in a mat, those cells are much more condensed. So the toxin loading can be, not always, but can be much higher. And some work out in New Zealand has shown it takes about 5 grams of tissue, so about as much as the end of your thumb, to be lethal. So knowing that those mats are what pose that risk, we just need to be able to avoid them. And as always, practice responsible stewardship. So protect our waterways from gray water and nutrients. This is for several reasons. Even if nutrients aren't necessarily the main driver of cyanobacteria in your particular region, we should always avoid nutrient loading, just in case we want to avoid future problems that may come along with that as well. Protecting the riparian zone, keeping trees intact, and minimizing disturbance to shorelines. And the last thing for responsible stewardship that I want to emphasize is clean, drain, dry. You've probably heard of this program through protection from the transport of invasive species. We still don't know how these cyanobacteria mats can move from one system to another. And it's possible that by taking material on boat or trailers, you can be inoculating another system. And these mats, when it's the season where they're floating up onto the water surface, they stick to boats and can be transported and moved around. My team carries a wash-off station to wash these off at the water's edge before they pull out. And you also don't want them on your rubber boots, for instance, walking home, walking into the house with them, because dogs are attracted to them and dogs will seek them out. And lastly, the thing you should do is exactly what I've been seeing, is continue to report observations of blooms to Department of Environment and local government. Even if there is nothing that they can do to sample that bloom at that time, that record is still meaningful for scientists trying to understand these blooms, and we can understand the history of them in certain areas. It helps us understand what is going on within a particular system. So, and with that, I just want to acknowledge my teams and collaborators. We've got the Aquatic Ecosystem Study Team, the National Research Council of Canada, Dr. Janice Lawrence Lab, and there's a ton of NGO partners here as well. Like I said, ACAP St. John is really helping us share this information, and all of the NGOs that have helped us collect samples or sent us samples and observations. We appreciate your eyes on the water. And with that, I welcome any questions.
That's great. We have some wonderful questions here. I'm going to start with the first one I received. Marian Langhus asks, I am the emergency response coordinator in the village of Gagetown, and I am wondering if we should have a procedure for cyanobacteria in our emergency measures organization booklet. So
procedure as in a reaction plan, or
yeah, what to do if you see something you suspect might be a cyanobacteria mat. I assume, Marian, that's what you're thinking.
Yeah, because Gagetown is downriver of an area that we know where there is extensive mat proliferation, wash up could feasibly happen. And I think that we have found, I haven't received all of the toxin analysis back, but we definitely found some cyanobacteria, smaller mats in that region. So it's possible that you could see more mat growth, especially with increased awareness and more eyes on the water. But the reaction plan should largely be communication outreach so people know to avoid them. And maybe communicating at peak times, like during the summer when you've got increased traffic on the water. It can be things like signage at boat launches, as well as whatever other communications you have internally within the village.
Sorry, go ahead.
As far as a reaction plan to treat it in any way, that wouldn't be feasible, but for communicating the issue, it's generally a good idea.
We had a very specific question from Gerard Suma asking, what do you think of using Phoslock to treat a lake experiencing blue-green algae bloom?
Yeah, so Phoslock is a substance that is thought to inhibit nutrient uptake and therefore slow the proliferation of the bacteria. I honestly don't know enough about the interactions of that environmentally. I can look into that. Sorry, what was the name?
Gerard Suma, and I think he indicated he is with, sorry, just excuse me a second while I scroll back through the names and their affiliations. Gerard, I think, is with, sorry, I can't spot it right off the bat. There's a lot of names.
That's okay.
We can get back to him. I can find it and get it back to you.
Yeah. So I heard of this. I don't know enough about the potential interactions. The problem with things like that, when we start making these large scale manipulations, they've often been studied in a very limited region and no two sets of environmental circumstances are the same. So there may be different environmental interactions with different compounds that have adverse effects that you can't anticipate depending on what you're applying it to versus what that's been studied in. And the other aspect is are we then controlling for recreation? So and maybe not the ecosystem, because what will that do to the other organisms within the ecosystem and the other bacteria that are an important part? So we could risk messing things up worse or just in a different way by taking this management strategy approach. But I again, my disclaimer is I don't know enough about the variability and how well that's been studied in certain systems. But I'd like to talk about it.
Yeah, so the blue-green algae really isn't cyanobacteria necessarily. So I guess the idea, you mentioned that even very clear water can be hosting these cyanobacteria mats.
Clear, pristine waters. So, yeah, and that's what they're seeing in New Zealand as well. We're not the only ones seeing that. They're seeing that in New Zealand as well. And mats have even been found in the Arctic in undisturbed waters.
Okay. A question from Heather Bursey asks, are there safer times of year to have your dog swim in New Brunswick waters? Good question.
That is an excellent question. Unfortunately, we have to be conservative. It's better to be safe than it is to be sorry. Knowing that I can see these mats growing early season, cyanobacteria can proliferate very early in the season, as well as later into the season. So we do have toxic mat samples coming from as late as October and November in years previous. So it's that they are still there. But if you can look around the area and you don't see any mats and you don't see any green plankton in the water, then you should be safe. Pay attention to your dog. Don't let your dog drink water. Bring water for your dog to drink, and make sure if it's nosing around the riparian zone or wash up, pay attention if it's interested in something and don't let it eat anything.
Okay, so I understand those mats can attract dogs, the smell of them on the shore when they're drying or somehow not in the water. Is that true?
Yeah, they attract dogs. I don't
Okay.
know why, what compound it is that dogs are attracted to, but that is why we've seen this in dogs. I will say that we have not observed any fish kills in the region of these mats.
Okay.
So we haven't observed any cyanotoxins that have caused fish kills in these
Okay,
regions.
that answers one question from Mike Rushton. Does it have a significant impact on aquatic species, invertebrates and fish, etc.?
Yeah.
I guess the answer is
So
no so far.
yeah, planktonic blooms in lakes tend to more so because the organisms can't help, other living things can't help but ingest the
Mhm.
plankton. So fish drawing the plankton, like planktonic cyanobacteria across their gills, for instance.
Okay.
That is a bit harder. So that's why we see more fish kills in lakes because of toxins.
Mhm.
But also anatoxin-a, the one that's killed the dogs in the Fredericton region, is considered to be unstable in water. It breaks down in water and with UV light. So that's another good part of the story. So it means that the biggest risk is the ingestion of the mat itself, where the toxin is. Toxins do leach into the water, but they often are not stable and break down.
Okay, so water helps them break down and UV rays as well. Okay, couple of other questions here. Heather Bursey again asks, is there a safe water in New Brunswick? I guess is there anywhere you can take your dog and it will not guaranteed not to see cyanobacteria? Sorry, did you not catch that?
Sorry, you're
I'm breaking up.
yeah, cut it. Sorry.
Okay, I'll try again. Is there a safe water in New Brunswick where there is known to be no cyanobacteria?
Cyanobacteria are ubiquitous in the environment, so you're never going to find a water that
Mhm.
doesn't have any bacteria, nor would you want to, because they are an important part of the ecosystem. I think what the question means is that there's no known toxic cyanobacteria, but there's a lot that we haven't tested. So I refrain from pinpointing an area and saying for sure it's safe when there's many areas that I haven't tested, and my work focuses, I'm drawn to the areas that are producing the toxins. So those are the areas that I'm studying.
I turned my video off, so hopefully that'll stabilize the connection a little better.
I do always refer people to the Government of New Brunswick lake advisories. Those are areas that are known to have produced cyanobacteria blooms that could be potentially toxic. Now, once the way that these advisories work is once a water body has been put on that advisory, it stays there. It is never removed. And the reason for that is because blooms tend to reoccur in areas where they've occurred in the past. So just because something isn't blooming at a given time doesn't mean that a few hours later it couldn't be. But it doesn't mean that everything on that advisory is constantly blooming either.
Okay, very good. So there's no real safe water. You have to use common sense and look at the water. Okay.
The water. Yeah, you should always be watching for
Okay.
the green color, which is the planktonic bloom, and the benthic bloom.
Alan Beck asks, is cyanobacteria specifically a freshwater concern, or is it commonly found in salt water as well?
I actually don't know how commonly it is in saltwater. I know that it is also in marshes. It is brackish, but I don't know that, Alan. I hear of other algal blooms, planktonic algal blooms in saltwater, but I've certainly not familiar with the issues at the scale that I study them in the freshwater.
Okay, I've heard of red tide, I think, affecting shellfish in saltwater, but I don't know if that's a bacteria or what it is actually.
That's an algae. That's a planktonic algae. It's a dinoflagellate.
Okay,
Yeah.
so it's a plant-based toxin. All right, so we have a couple of other questions. Has there any research been done on Macrophyte Lake?
No.
No, okay. That's an easy one.
Not. Well, my colleagues at CRI, I have not done research on Macrophyte Lake, but my colleagues at CRI, I think actually did a plankton tow there, I want to say 2017. I have done. Does Macrophyte have a history of blooming as well, planktonic blooms?
I don't know.
It's my understanding that that is a very tannin-loaded lake and it's very dark.
Again, I haven't boated there, so I don't know. Our home is on Grand Lake, where again we have quite a few tannins, but it can be very clear at times as well. So
Okay.
I don't know about Macrophyte. It's shallower than
Yeah.
Grand Lake. I know that.
If I find out who studying blooms at CRI before me, the planktonic blooms. So I've been emphasizing the benthic blooms.
Mhm.
And if there's any work that's been done, I can share that in that direction.
Okay, thank you. Debbie Peck asks, would the agitation associated with water flowing through the Mactaquac Dam have any impact on breaking up floating mats of cyanobacteria and helping spread them in the downriver area?
Yeah, we will be looking at that, but beyond just the perspective of the dam regulation, we think about like what would happen in that region if that dam wasn't there. And in summer, when we see these mats proliferate, it would be an area where there are very, very low flows. And then with rainfall, you would naturally have flushing events and disturbance anyways. So what the relative impact is of the dam influencing flows there, or just the natural bathymetry of the area and those natural flows, we don't yet know, but we are looking at how flows influence this cyanobacterial mat proliferation.
Okay, so it's an open problem, I guess, still being explored.
It is. It's part of the habitat question. So
Mhm.
part of how much disturbance and what type of environmental flows do these organisms thrive in and tolerate, and what causes them? What are those thresholds when they mature and start to wash up and move?
Okay, we have a question from Gordon Wilson. What are the major contributors to producing the benthic mats, I guess, the cyanotoxins? And ultraviolet rays, have ultraviolet rays hinder or help them produce more, or can they kill them? We've heard, I guess, that they can kill them. And are there any positives that are nature's way of cleaning water systems with cyanobacteria?
Any nature's way. So to the nature's cleaning system, I'm going to say with no, because what happens is these bacteria, when they come into a stressful nature condition that causes them to die back, so maybe it's too much heat or extreme temperatures or totally cleansing flows. I've seen these mats totally removed, scoured so well that you would never know a mat was there, clean rocks.
Mhm.
But they produce a resting spore stage that it goes down into the sediment. So even if nature has an environmental condition that is a temporary solution, it's a temporary solution. And then they tend to come back. As far as the question about ultraviolet light, these organisms are well adapted to thrive in a range of light conditions. They need sunlight, but they've also been high and dry on shores and then resubmerged. So they go through exposure and submersion gradients. And it's more the lack of light that is challenging for them than the abundance of light.
Mhm.
So nature's way of cleansing them, it does happen. It's cyclic. Some years we've seen mat proliferation worse than others. And I think it's got to do with the shear stress of water velocities, purging them at a certain time and then them struggling to come back. But what the exact environmental conditions are around that, we still don't know.
Okay. Mike Rushton is asking, a major hurdle now is slow turnaround time for sample analysis. So do you think that test strips or test tubes, a faster way of doing analysis, would be possible? Is it an alternative to what's done now? I guess it's done in the microbiology lab with genetic analysis.
It is. And so, yeah, it does take a long time. The thing is about any of those fast rapid tests that get developed, you're specifying one type of toxin that you're targeting, and there are hundreds of different types, and they can be produced simultaneously. So you're not going to have a one strip answers all.
Okay.
At first perspective, to do the level of analysis we did on toxins, we had access to the biotoxin lab in Halifax. Hiccups. And they were able to take, at the start of the study, 16 samples, and it was going to take them the better part of the year to be able to process them, just so we could identify. That's how involved toxin analysis is they have millions of dollars worth of equipment and numerous trained technicians with PhDs that operate this lab just to be able to identify different toxin types. So that's the highest resolution toxin analysis you can do. Then while we were doing this study, they got a new machine and we did like 150 samples, but they couldn't open it up to the general public because they don't have the capacity. And this is the only lab that does this type of analysis. So the best approach, especially considering the variability of toxins in the environment, is to pretend or act like things are toxic and avoid interacting with them. That's the simplest, safest approach, because that's really the only way to guarantee. Okay,
so there's no quick, fast test now that can identify, especially the toxic cyanobacteria.
Yeah.
Okay.
Unless you're targeting a very specific type of toxin, but if you were, you would already know what was a bloom characteristic of that area. So it wouldn't really tell you anything else.
Okay.
But for new blooms, toxin testing toxins, I think you will see capacities expanding, and maybe some NGOs will be able to submit some more samples within the next year or two at UNB if everything goes as planned with the new lab setup, but it may not be as robust as we're hoping for. Everyone wants a safe answer to know that something is not toxic, but even a negative sample can't guarantee that for your watershed.
Okay. We have a question from Kirby Morrill. He asks, can the toxins be absorbed on contact or only by ingestion? And are the species that produce harmful toxins ubiquitous, like everywhere? Or would there be any point in eDNA sampling for presence absence of particular species as a risk indicator? So questions I don't really know anything about.
Yeah. Hi, Kirby. First of all, thanks for tuning in. So the multiple questions here. First, eDNA with your species targeted question. The same species can be toxic or not toxic depending on its genes. And because these are bacteria, they can pick up toxin genes from their environment, from other organisms. So that's how they can become toxic. So eDNA is a bit harder. And even if you had a positive eDNA sample, it wouldn't tell you anything about the amount of toxins. So part of the issue with an analysis is just because you can detect it doesn't mean that it's biologically significant. And so that's really what we're concerned about with the mats here, is what are our toxin loads? How much toxin are we looking at? The World Health Organization is starting to release more and more limits for cyanotoxins, for advisories for intakes and for exposure. And that is inevitably going to be the most important information for guidelines, because it's about the thresholds of toxins that we're concerned about. With regards to, what was the first part of the question?
Are they absorbed on contact? Can the toxins be absorbed on contact or only by ingestion?
Right. So the three types of toxins I said were the hepatotoxins that affect the liver, the neurotoxins, the neurological system, and the dermatoxins. The dermatoxins that are the irritants are the ones that irritate the skin. So those are the toxins that when you have contact, you have some effect. The hepatotoxins that affect the liver and kidneys, those ones are usually concerns for things like drinking water managers, for reservoirs, for long-term chronic exposures. And then the neurotoxins are the acute toxins, the fast-acting ones that can result in death within half an hour, and they require ingestion. So the first two types of toxins, the hepatotoxins and neurotoxins, ingestion is a concern, and the dermatoxins, the irritants, that's the contact.
Okay, that's clear. Thank you. One other question from Dinah Stalker. Is there an effective way to best treat water drawn from the lake that's going to be used for showers? Could you filter it out somehow, or treat it such that, put it under a UV light or something that would get rid of cyanobacteria or at least kill the toxic ones.
Even with things that kill the organisms, it doesn't necessarily mean that you kill the toxins. And without knowing the toxins that are in that sample or that water, it's hard to know how to treat. So for instance, anatoxins would naturally break down, microcystins not so much. So that would really depend on what specifically it was you were dealing with. With the terms of the Washtenamock Lake, I will say that I have seen blooms there. I still swim in there. I plug my beer when I jump off the sailboat so that I don't ingest the water.
Okay.
And so I wouldn't worry about that because you're not ingesting the water. Don't maybe shower with your mouth open and drink at the same time, and you should be good. So, and if something was to change, if we had reports from a dermatoxin, then that may differ. But as far as I know, in the Washtenamock Lake, there has been, I think Janice showed some evidence of microcystins, which would be expected with the blooms that happen in the lake. But as far as showering, you're not ingesting the water.
Right.
So
Okay. So at least in Washtenamock, it should be okay just to use the lake water to shower. As far as you know. Yes.
I have
Okay.
not stopped swimming in the Washtenamock yet, and
Okay.
I hope that day never comes.
Yes. Okay. If there is a bloom, Sarah Hart asks, if there is a bloom, can the toxins become airborne, particularly on cool, windy fall days when there's mist coming off the water?
There are some types of toxins that can become airborne. Anatoxin-a, this one that is considered to be responsible for killing the dogs, is not thought to volatilize. As far as I know, we don't have any toxins or any reports of toxins in our waters that have reached a high enough limit where there have been volatiles. In other areas for specific toxin types, they have shown that. There was a situation, I think it was Florida, where researchers in the lab were actually growing these organisms to study them, and they volatilized them in the lab. And it can also, I guess, theoretically happen in the environment, but I don't know of any evidence of that happening in our.
Okay, so natural environments, the probability, as far as you know, is low for that happening, becoming airborne.
Yeah, it would depend on the toxin type too, and certainly for the mats that we're studying in the Fredericton region, the anatoxin-a that has not been considered to volatilize.
Okay. Question from Jennifer McPhail. She asks, you mentioned a new study looking to develop microprobes. Could you talk a little more about that? What are they testing for? A specific bacteria strain or specific toxins?
Right, so that's my colleague's research, not mine directly. I'm partnering with her on the field study, but to briefly summarize, she has microprobes which can test things like pH, oxygen, phosphorus, nitrogen, at the level of, I think it's like 1/10 of a millimeter, maybe even finer. And so what that means is we can look inside the mat and look at those geochemical cycles to see what it's doing. This is stemming from work that happened in Susie Wood's lab in New Zealand, where they found that nutrient availability within the mat was 300 times higher than outside of the mat because they have their own chemistry that can change the nutrient availability. So this is why these mats can thrive in some areas that are naturally nutrient very low. So is that something that's happening in our mats? And that's what my colleague Allison will be examining.
Okay, so that's clear. Robert Powell asks, have you tested round mill cove on Grand Lake? That might be an easy answer. I don't know.
I haven't, no.
Okay. All right. Has there been any cyanobacteria found in the main Miramichi River from Heather Bursey?
I'm sure there has been cyanobacteria found, but we have not found. I do have colleagues studying there extensively for the collaboration of Atlantic salmon tomorrow. And they have seen my mats through presentations like this, and they have not come back to me and said, hey, we're finding something like this in the Miramichi.
Okay, so no evidence so far that you're aware of.
No evidence so far.
Okay, and Yoho Lake? What about cyanobacteria in Yoho Lake?
Yoho Lake does bloom. We did a plankton tow sample there in 2017, I think.
Mhm.
That was not a benthic bloom, but a planktonic bloom. And I think it was largely determined to be an Anabaena bloom. No toxin analysis was done at the time, but Anabaena is a type of cyanobacteria that does have the potential to produce microcystins, at least, and maybe some anatoxin-a as well.
Okay. Toxins harmful to the fish habitat. Are the toxins harmful to the fish habitat? I guess you mentioned the fish itself would draw cyanobacteria over
If
their
you've got
gills,
fish
but
enclosed lake and there's a high density bloom, then they can't avoid it. But like I said, we've had no evidence of fish kills at our sites. And in fact, one day I was in the water and a salmon, most curious salmon I'd ever seen, was swimming all around us, perfectly healthy. And we've seen fish jumping now and then, but no dead fish washing up on shore. And even if we did see dead fish washing up on shore, first of all, it would have to be a lot. And there are natural diseases that run through fish populations that cause fish kills as well. But we have not seen anything that we would attribute to cyanotoxins.
okay. Sharon is asking, we have green algae on the shore that turns brown, like in your picture.
Yeah.
Is that considered toxic? I guess you wouldn't know probably, but it's maybe.
I wouldn't know, but what I would encourage Sharon to do is to collect a sample, send it to me. I'll throw it on the microscope if it's the same type. So I didn't show you guys the microscope identification side of things today because you guys probably don't have microscopes at your house. So I was showing the very general appearance. And what I would do is I would look at it and see if it's a potential toxin producer. And if it is, I'll see if Janice has capacity to screen for toxin genes. And we can see also.
So you're saying collect the sample and send it to you? Is that the possibility?
Yeah.
Oh.
Yeah.
Okay.
I can't always guarantee how far I can go with analysis. Again, that depends on capacity, but I will, anything, especially if it's a new region where we don't have any idea of what's going on, I will take a look at it, see if I can identify at least what type it is. I may not be able to identify whether or not it's toxic, but I might be able to tell you whether or not it has been a toxic producer elsewhere. Also, record pictures.
She says she's on Macrophyte Lake. She just retyped in on something else here.
Yeah. Also, yeah, record pictures if you can.
And the sample, does it need to be cooled? Like when we were taking water samples for analysis, should you cool the sample, or does it need to be in a baggie?
I just put it in a jar and put it in the mail because they're warm often when we collect them, right? When I'm collecting my samples in mid-July, that water is 20-some degrees. So if I throw it in a cooler, then I'm stressing those cells. Right. So I will just actually put river water in the cooler until I get back to the lab. And then we do freeze them quickly once we're in the lab, but we don't stress the cells. So
it should be the same day transmitted. I mean, she's at Macrophyte Lake, it seems, so could they drive it to Fredericton the same day? Would that be the best approach? If it's in the mail, it might be, who knows what would happen to it.
I've received specimens in the mail that I can still tell what they are. Yeah.
Okay. So could I share the address then to send them to? Would that be a possibility then after this is done?
Yeah, they can, as long as there is the disclaimer that it's whatever I have the capacity to analyze at the time. So I will save them up, and sometimes it takes some time to get to them, but I only ask that people also just send me an email to give me a heads up that it's coming in, and then I can continue the conversation with any details that I need. For instance, where was it collected? When was it collected?
Yeah, so you do need identification of where and when, and picture would be good too when they actually.
And again, I may not be able to do any toxin analysis. That doesn't actually happen in my lab, but if I have a specimen of interest, for instance, if we see a lot of it growing at Macrophyte Lake, it's covering a large area, and I have had some samples that I haven't fulfilled with my collaboration with Janice, if I might be able to say, oh, let's do this sample instead of the one we planned. So there's a few samples missing from this year because our mats actually washed off very early and didn't reestablish very well. So I have a little bit more room for a couple more samples.
Okay, so perhaps we shouldn't just send out a general invitation, but maybe some that have specific questions. Like I think I could get Sharon Cormier's email address and maybe give her the details.
Yeah.
Okay, all
Yeah.
right.
I
I don't
would
want
love
to
to-
overload your team with like thousands of samples that are just going to be discarded, because that's sort of a,
So it's not really in our like capacity to take on samples beyond what our research samples are, but I see the value in working with these groups that have more eyes in the water. I can't be everywhere at once. And this is how we find about other interesting things in other areas. And it's important. And there is no other place in New Brunswick where we can do these types of things. There is no sample facility. Even RPC can't conduct these types of analysis. So I understand how it's frustrating for people that are wanting to start to figure out what's going on to not have any answers.
Okay, well, that at least we thank you very much for offering to at least identify if it's algae or cyanobacteria that is being looked at. You know, that's a big one right there, I think. Can you do that in the microscope? Just see whether it's actually green algae or
the bacteria cells are much more smaller than the green algal cells. There's other characteristics of the cells in the type.
Just that knowledge alone would probably be helpful for a lot of people, I think. That's just me surmising that. Okay, that's all the questions I see from the audience, from the participants. I wanted to ask one question myself about signage at boat launches. So, oh wait, Dinah Stalker just sent a message that just came in anyway. Are there tests to determine if a dog is ill but did not die from exposure? In other words, that they had a past ingestion of toxic cyanobacteria, but now they seem better.
Not that I'm aware of. I do have a friend that's a vet, but the vet, when we had our toxin discussion, we acknowledged that there's nothing even they can do if the dog gets to the vet after having ingested the toxin. So the way that they identified the toxin from these dogs that died is actually had stomach contents. They actually had algae from the stomach, this mat material, which they did two things. They analyzed the chemistry of that and they regrew those cells. So they took those cells out of the dog's stomach for things that it eaten and then put it in culture and grew it in the lab and saw that it was a type of toxin producer and produced toxin. So that was pretty affirmative that it was indeed anatoxin-a. But what I don't believe exists is any type of blood test to detect the toxin or anything like that. So there is no test that I'm aware of unless it's been developed recently. And there's nothing that they can do if you get your dog to the vet. So this is why you have to avoid.
Okay. Yeah, the one question I wanted to ask was about signage at boat launches. You said maybe check for cyanobacteria. Is there any kind of a standard signage that's been developed by your team or others you know of for cyanobacteria, watch for cyanobacteria, or how to identify it?
I saw a great one that Department of Environment and Health had done, and it's put up at Hart's Island. So maybe contact them and ask them what they had done or if there's access to that sign. The reason that they chose Hart Island RV resort is because that was an area where mats had washed up and dogs had encountered them. And it's a high traffic public area for boat launch. But if that sign has already been designed, then maybe there is an opportunity for groups to do more.
Thank you very much. That was Department of Environment and Local Government, you said. Yeah. Okay. Great. I don't have any other questions and I don't see any others in our list. So thank you again very much for helping us out by spreading the word about this issue of cyanobacteria toxins and how to identify them. A lot of good information, and I should have the video from this webinar available on our website in probably 2 weeks. It should be up, and I will send everybody that attended a link for that video.
Okay, great. Well, thank you everyone for attending and thank you for organizing. Like I said, it's really nice to have somebody help with that, to help share this information. And I'm sure that I'll hear from some of you and I look forward to our conversations and we'll continue to get the information out there as needed.
Okay, thanks again and good luck on your teaching efforts this fall.
Thank you very much.
Okay.
Take care.
Good night, everyone.